Hello, everyone. Thank you all for joining us for our September Research Spotlight webinar. I hope you all had a lovely summer. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. To start, I want to thank our sponsors for this webinar, Ionis Pharmaceuticals, Cleveland Clinic Laruvo Center for Brain Health, Novartis, and Argenta Therapeutics. Also, a big thanks to everyone who supports the HDF. It is because of you that we are able to fund exciting science that is transforming what we know about Huntington's disease. And of course, a big thanks to all of you for being here. Today's speakers are winners of our 2024 Transformative Research Award, which is one of our annual funding mechanisms that gives $1 million over 2 years to collaborative teams for projects that will advance us toward therapeutics for Huntington's disease. Our speakers today are Dr. Andrew Yu, who is a world leader in stem cell research and is the one who developed a direct conversion protocol that turns skin cells directly into brain cells. We're also joined by Dr. Osama Dalahmah, who is a neuropathologist and a renowned expert in brain structures and the effects of HD within the central nervous system. Doctors Yu and Dalahmah have identified bits of genetic message that are only present in people with HD. These genetic bits are called non-coding RNAs. They're pieces of genetic material that the cell never intends to turn into proteins, but they themselves perform regulatory functions throughout the cell. And 2 such non-coding RNAs have been identified by this team to only be expressed in people who are symptomatic with HD. And so this is exciting because it suggests that the presence of these molecules is related to the disease itself and not just the genetic presence of the HD gene. And this is what makes them very attractive as therapeutic targets, which I know we are all eager to hear about. But before I pass it over to Osama, I want to remind you all you can ask questions in the Q&A box throughout the talk. So try to remember to use the Q&A box, not the chat box. We should have about 15-20 minutes at the end for a Q&A. So type your questions in as you think of them, and we will get to as many of those as we can at the end. Also, a reminder that this will be recorded and available on our website and YouTube channel in about 2 weeks. So if you want to watch it again or share it with someone, it will be available for that. Thank you again, everyone, for being here, and I will pass it over to Osama.
Thank you, Sarah. So today, I am excited to tell you about our work, our collaborative work, using human brains and neurons to transform Huntington's disease research, which is a collaborative project between Columbia University and Washington University in St. Louis with Dr. Yu's lab. And next slide, please. I just want to start by introducing my lab. We're a group at Columbia University, and I'd like to thank the people, the researchers, the students, the scientists in my lab who made this work possible, of course, with support from the Hereditary Disease Foundation and with precious gifts from people who lived with Huntington disease and donated their brains for research, which allowed us to do this research project. Next, please. So a brief introduction about Huntington disease. As you may all know, Huntington disease is an autosomal dominant disease. It's caused by a mutation in the Huntington gene. And this mutation is passed on in a dominant manner. That means that you only need 1 copy of the gene to get the disease. Typically, the mutation has accumulation of these repeats, repetitive sequences in our DNA, in a specific part of the Huntington gene that leads to an accumulation of these repeats. And after a certain threshold of repeat accumulation, people become symptomatic. And one of the regions in the brain that is affected in Huntington disease is the basal ganglia. And that's part of the brain that helps us control our movements and coordinate our movements. And what ends up happening is that the mutation causes a lot of glutamine residues. Think of them as sticky stuff in the protein that allows the protein to accumulate and aggregate. And in the practice of neuropathology, we see protein aggregation when we see neurodegeneration. So generally speaking, in normal people, we don't see Huntington protein aggregates. In people with Huntington disease, we see Huntington protein aggregates. Next slide, please. This is just a schematic showing you how the protein that with the mutation can accumulate. And we have tools to look at neuronal loss and nerve damage that happens to people with Huntington disease using imaging techniques like magnetic resonance imaging and CT scans. And if you look closely at this image, you'll find that a certain part of the brain that controls the movement, I referred to it before as the basal ganglia, is severely degenerated. It's severely affected. Next slide, please. And this is what we do in neuropathology. We do brain autopsies. So when the ultimate gift is given and patients or people with Huntington disease donate their brains to research, and other people who actually don't have Huntington disease also end up donating their brains for research, we're able to look at the changes that happen in the brain as a consequence of the disease. So we can investigate the disease pathology in more details. So on the right-hand side, this is a slice of the brain at the level of the basal ganglia on the right-hand side, where we see a control brain. That's the brain of a person who died without Huntington disease. So you can see that the basal ganglia region, which is in the center of which the pointer is pointing to it, that's the center of that slice that controls the movement in this nervous system, is not degenerated. But if you look on the left-hand side, where we see a slice from a patient with Huntington disease, you see that that part of the brain is severely degenerated. It's atrophied, and that corresponds to neuronal loss. Next slide, please. So we can use classical traditional techniques to study uh, the pathology, the changes that happen in the brain using microscopy. So we can look under the microscope and look at the neurons, which end up dying. And we see that even within the same brain region that is severely affected in Huntington disease, we see variability in disease severity. So that region outlined in red is called the caudate nucleus. As I mentioned, it controls or regulates our movements. Um, has an interesting feature whereby if you look at the upper part of the slide, next transition, please, you will see that a lot of the neurons die. Next, please. But if you look at the lower part of the caudate nucleus, next slide, please, you will find that there is less severe neuronal loss and degeneration. The blue pigment here, or the blue color, indicates the nerve projections. So when neurons die, their projections degenerate and are essentially eliminated. So in the lower part of the caudate region, you see a lot of these uh, blue projections, which means that the neurons are alive. But in the upper part of the caudate nucleus, we see that these blue regions are essentially gone, which means that the neurons are dead. Next slide, please. Using traditional techniques, we can also look for protein accumulation using a technique called immunohistochemistry. And here we have to look really hard to find the protein aggregates. And in using this technique, whatever is brown is labeled. So here we use antibodies that detect specific antigens or specific proteins. In this case, it's Huntington disease, Huntington protein. Next transition, please. So this cell here, which is a neuron, has a brown dot in the middle. Next transition, please. And if you zoom in, you see that there is a brown dot in the middle, and that is Huntington protein aggregates. Next slide, please. However, as Sarah mentioned, not all genes in our DNA make proteins, but we can look at the messages that these genes make. and the messages are called RNA. There's a technique called RNA scope that allows us to measure these messages, measure the RNA. So for the genes that do not make proteins, we have to look for them using this technique, RNA scope. And on the right-hand side, you see a bunch of nuclei in blue and a lot of dots in different colors. Each dot represents one message, and each color represents a different gene. So using this approach, we wanted to investigate the presence of what we call HD link. Next slide, please. So what are we looking for? We're looking for a signal in the human brain, a non-coding RNA that increases in Huntington's disease. We call it HD link. Next slide, next transition, please. This is a depiction on the top right of what it looks like, how it is, how scientists think about it. And on the left-hand side, I'm showing you a part of the brain, the caudate nucleus that I kept going back to, and we use the technique called RNA scope that I just introduced to you to quantify the presence of that message in the neurons in Huntington disease. And those are represented as these green dots. And if we quantify that in the caudate nucleus, we see that there's a significant increase in this message in the nuclei and neurons of Huntington disease donors compared to people who died without Huntington disease. And this is quantified in the diagram in the middle. And we can also measure that, as Dr. Yu did, using RNA sequencing, where we actually measure the actual sequence of the messages and figure out what each message is telling us. And we can figure out if that message is HD link or it's not. And in this case here, Dr. Yu sequenced three donors who didn't have HD, and three donors who had HD, or two donors who didn't have HD, and three donors who had HD, and was able to detect the presence of this HD link in the HD donors, but not in the control individuals. And that was done from that can be detected in neurons that Dr. Yu can differentiate from skin biopsies from living people with HD. And Dr. Yu will tell you more about this important gene.
Thank you, Osama, for the great introduction, and then sort of leeway into this gene called HD link. Before I describe what HD link is, I'd like to first introduce my lab, where we've tried to focus on various aspects of molecular mechanisms that underlie the onset of neurodegeneration. And then in terms of modeling diseases, my lab primarily focuses on modeling Huntington's disease and Alzheimer's disease. And for today's talk, it'll be mainly based on the findings that were made by exceptional graduate student in the lab, Ji-Sun Kwon. She's about to graduate, and then kind of have a brief description of her findings so far. So as Osama briefly mentioned, and also Sarah briefly mentioned, my lab focuses on the development of what we call cell-fate reprogramming approaches. And what I mean by that is we can culture non-neuronal cell types, for example, human skin fibroblasts in culture dishes. And we have a way of turning those cultured fibroblasts directly into neurons without going through the canonical stem cell stages. And this is what's called direct neural reprogramming. And my lab specializes in the use of genes called microRNAs, especially microRNAs called miR9 and miR124. So what these microRNAs represent is in the context of neural development, these are microRNAs highly expressed in neurons of the brain. But when we topically introduce these microRNAs into skin fibroblasts, we have a way of turning this skin fibroblasts directly into neurons. So we call this process microRNA mediated direct neural reprogramming, or sometimes we call it conversion, because we go from fibroblasts directly into neurons. And the reason why we do that is mainly for the purpose of modeling kind of late onset or adult onset neurodegeneration phenotypes in cultured dishes, in cultured patient-derived neurons. So here's a couple of examples of what the whole process looks like if we actually look at the cells under microscope. So in this starting picture, you see a bunch of cells that have been plated as fibroblasts. And when cells start expressing these microRNAs, from now I'm just going to call them miR9 and miR124, so 9124. You can see the cells actually over time changing their morphology to what appears to be morphologically what appears to be neurons. And as you can see, the process is really one cell to another cell type conversion. And this conversion needs to be very highly efficient because if it's not efficient, then we can't really model the disease. So another way of looking at this is to use what we call fluorescent reporter. For example, this reporter called RIP, which in fibroblast population, you will not see a lot of fibroblasts expressing this reporter because the cell identity is within the fibroblast context. And over time, with this microRNAs being expressed, what you can observe is that in this field of view, you'll see emergence of cells expressing this reporter expression because cells are actually being turned into neurons. And then in terms of modeling the disease, what we also have to care about is what types of neuronal subtypes that we can generate. Mainly because in different contexts of neurodegeneration, different neuronal subtypes are differentially affected. So for the importance of today's talk, what my lab works on is to generate striatal neurons, and especially striatal medium spiny neurons, which is the primary target of Huntington's disease. And the way we go about doing that is to combine these microRNAs. And what these microRNAs do is when they go into fibroblasts, then they induce the plasticity of the cell fate, but more in terms of pan-neuronal plasticity. And then what we can also do is we can drive this microRNA-mediated neuronal reprogramming process into very distinct types of neurons. And the way we go about doing that is we combine these microRNAs, so we call this microRNA-induced neuronal state, and combine that with the different transcription factors, differentially expressed in different parts of the brain. So by doing that, what we do is then we combine miR9-124 with these transcription factors called the CTIP2, DLX1, 2, and MIT1L. When we do that, then we basically instruct the neuronal conversion process distinctly into striatal medium spiny neurons. And then some of the colors are what we call them markers of medium spiny neurons. If we combine microRNAs with a NeuroD2, ASL1, MIT1L, then what we do is then we sort of push the neuronal conversion process into cortical neurons. The reason why we do this is because when we convert the cells from fibroblasts directly into neurons, and because we don't go through the canonical stem cell stage, but instead we induce the neuronal plasticity, but at the same time, we basically alter the state of cell identity from fibroblasts into neurons. So by doing that, we basically carry over the age information that was stored in pre-reprogrammed fibroblasts into reprogrammed neurons. And that really allows us to generate human neurons that mimic the age of fibroblast donors. And then this part becomes hugely important in terms of modeling sort of the late onset aspect of neurodegeneration in different disease contexts. So the reason why we care so much about driving this neuronal conversion into very distinct neuronal subtypes is because by generating neurons that are vulnerable to certain disease contexts, we can actually model the neurodegeneration process. So here's one example where we combine microRNAs, and as I mentioned, CTIP2-DLX1-MIL1L. We can push the conversion to striatal medium spiny neurons, and if we look at them over time in culture, this is what the cells look like. And here are a couple of examples of what we can recapitulate in tissue culture context, coming from the neurons differentiated from the fibroblast from the individuals with the Huntington's disease. So here's one example where we mark the cells, reprogram cells with certain markers. In this example, it's called TOP3. And the other examples meaning is MAP2, which is also a neuronal marker. But at the same time, when cells are turned into neurons, we can actually mark those cells that have accumulation of Huntington aggregation that you see in red color. Another important phenotype that we can monitor in this reprogrammed neurons is when cells acquire the neuronal identity over time, or what we can also see is cells degenerate in culture. And this is mainly because of the cells are coming from individuals with Huntington's disease, and they have mutation within the Huntington gene. So over the course of last 5 or more years, we demonstrated that we can model the adult onset neurodegeneration of Huntington's disease. And then this concept can be also applied to other disorders where aging plays a critical role. One prime example of that is sporadic late onset Alzheimer's disease. So in this case, we turned cells into neurons, but into cortical lineage of neurons, then we can actually capture the typical neuropathological features of Alzheimer's disease, including A-beta deposition, tau dysregulation, and actually the neurodegeneration that happens in reprogrammed neurons. So with that, the way we identified this gene called HD link is mainly because of it's mainly through transcriptome or gene expression analysis when we compare genes coming from reprogrammed neurons from healthy individuals versus Huntington's disease individuals. And then what we identified is that this gene is called link RNA. So link RNA basically stands for long non-coding RNA transcript, whereas typically RNAs can be translated into protein product, but for link RNAs, it stays as RNA transcript. So although it's not associated with the protein transcript production, but instead it affects like various biological processes, and some of those examples include chromatin regulation. Other examples could include microRNA function and gene expression. But the important thing about this gene transcript is that it's a human specific, so mice don't have it. So it kind of gives us insight into this human neurons, and when they express this transcript that is specific for the individuals with the Huntington's disease, we can actually study what this expression means in the context of HD reprogrammed neurons. So typically, this is a couple of examples of what how we analyze the gene expression patterns when we generate neurons or MSNs, medium spiny neurons, from Huntington individuals or control individuals. And over the time course, like we do this type of gene expression analysis like a lot. Every time we do this type of analysis, this gene, HD link, is one of our top genes. Like it really appears to be specific and robustly expressed in patient-derived neurons. So these are a couple of examples: HD reprogrammed neurons, control reprogrammed neurons, and you can see the expression of this gene is pretty specific for Huntington's neurons. Another example from different cohort of individuals compared to control. And also what's important is that this is an example where we generate neurons from individuals with Huntington's, but at a symptomatic stage. But if we compare the expression of this HD link expression to reprogrammed neurons from fibroblasts, but when the fibroblasts were collected before the onset of the disorder, HD pathological disorder. So these are typically younger individuals. And then if we compare the expression of an HD link, the expression appears to be more pronounced in HD neurons that were reprogrammed from fibroblasts coming from symptomatic individuals. So when we detect gene expression differences compared to control reprogrammed neurons, this difference could mean two different things. One is when there's a gene that goes up, it could be part of the mechanism where the diseased neurons are trying to compensate and trying to survive by activating certain processes. And another possibility is that when patient-derived neurons are expressing a certain message, it could mean that it's part of the pathogenic process driving neurodegeneration. So one good way of testing this idea is basically when the cells have this message being expressed, we try to repress it. So for repressing, we use this technique called short hairpin RNA with a small RNA components that's specifically targeting HD link expression. And if we do that, what we have seen is that cells when HD MSNs are repressed in terms of HD link expression, cells appear to be happier. So this kind of tells us that this HD link expression is not really compensatory mechanism, but it's part of the process that drives the neurodegeneration, the onset of neurodegeneration that we can measure in patient in HD individual based neurons. So knowing this, the role of HD link in terms of neurodegeneration, what we are really trying to understand is what's the molecular mechanism underlying why when HD link goes up, why cells are not as happy. Also, in the previous slide, in terms of repressing the expression of HD link, I said I'm using we are using this shRNA, but kind of in terms of therapeutic or in terms of the translational angle, I would like to test it, the expression by different modalities. So one way of going about doing that is to use antisense oligos against HD link. So it is a work in progress. And then we're really trying to understand with Osama how the HD link expression patterns differ in different disease grades and also in different brain regions. So that's what we have so far for today's talk. And I'd like to briefly acknowledge everyone where we are deeply grateful for any tissue or brain samples that we can acquire and study, especially for fibroblast samples that come from many different individuals with the Huntington's. Those fibroblast samples really allow us to go after these disease mechanisms using the individual specific neurons. And also I'd like to thank HDF Transformative Research Award because this award really allowed us to go after kind of the questions related to this disease-specific transcripts. And we hope that by really understanding the sort of molecular mechanisms associated with this disease-specific transcripts, we may be able to open different ways of thinking about how we can go about translational approaches for Huntington's disease.
Awesome. Thanks so much, guys. So before we start the Q&A, I just want to remind everyone, so keep submitting your questions in the Q&A box while we go through the Q&A session, and we'll get to as many questions as we can. So there's one question first that I will answer. It's not related to your talk, but someone is asking if we know when AMT 133 or data is going to be shared from Unicure. That has not been made public yet, but we are expecting results before the end of September. So very shortly, very soon would be my answer to that. And then there was a question, Osama, that came in around glial cells. It was more of a comment, but maybe I know you've extensively studied glial cells, so maybe you could talk a little bit about your work with glial cells in HD and how they're affected.
Absolutely. Yes, glial cells are affected in Huntington disease as I described. The mutation is in all brain cells, and glial cells make a large component of the brain. And together with a group of Leslie Thompson at UCI, we've discovered that glial cells are affected in Huntington disease in the sense that they lose their maturity. They're unable to mature and become functional, and that is seen at early stages in Huntington disease and in different brain regions. And one of the things that we do in the lab is to study astrocytes. If I'm gonna comment a little bit more about that, I would say that astrocytes also are affected in Huntington disease. We see Huntington protein aggregates in astrocytes, and astrocytes are the major support cells in the brain. They are the cells that make sure that every other cell is happy. It's like the most important cell in the brain, if neuroscientists will not contest that. But they do become dysfunctional in HD, and we found specific astrocyte states that could be beneficial. And we think that we have identified astrocyte states that could be deleterious, and we're studying that actively in the lab.
Thanks, Osama. So there's a lot of questions kind of about the involvement of HD-Link in HD. So this one asks, do you think HD-Link causes or drives HD pathology, or does HD-Link increase once HD begins?
One of the current ongoing experiments is to test whether overexpressing this or introducing this HD-Link in control neuron context, and then whether that will be sufficient to drive a neurodegeneration or not. And then also we can basically do the similar experiment at the same time, where we overexpress this link RNA in HD neuron context, whether we can make the disease phenotype worse. The first experiment where we overexpress in control neurons is an ongoing process, so we don't have the answer yet. But for HD neurons, where we overexpress even more beyond what's been also being expressed, that makes the neurodegeneration phenotype worse.
That's interesting. Thanks. This is kind of a similar question, so I'll throw it back to you, Andrew. But they ask, at what stage of the disease do you see HD-Link expressed? And this was a similar question I had. You mentioned that it's only expressed once people start to show symptoms, but does that correlate with a specific symptom? Like, does it correlate with the onset of chorea or depression? Can you kind of time where you start to see expression of HD-Link?
Yeah, it's a great question. So those samples that what I called pre-HD MSNs, they came from fibroblast samples reprogrammed, but fibroblast samples were from pre-symptomatic stage, but they're mostly in their teens and 20s. So I just, we have this very limited lifespan, because we don't have like longitudinally collected fibroblast samples from the individuals with the Huntington's. So it's kind of hard to answer in this, at least in the experiment context. So what we decided to do was because Osama is doing that sort of similar expression profiling analysis in brain samples, because he has like multiple grade brain samples available. So I think that's sort of what Osama is focusing on right now.
So to just comment on that, we see the expression of HD-Link in the brain samples from people who died of HD at the early stages of neurodegeneration. So it appears to be present when the neurons are still viable and the neurodegeneration is still early. But the way I think the importance of what Andrew's saying is that is the following. At brain autopsy, this is the end stage. We know where we can get a snapshot of what has happened over the years. But we don't have longitudinal samples. We have a snapshot at every stage of, say, or every disease severity grade. What would be extremely helpful is to collect fibroblasts and skin biopsies for Andrew to generate a repository from patients or from people who have HD and track the accumulation of this HD link as the disease progresses. So these paired samples, these longitudinal samples will allow us to answer this question. But as far as the human brain is concerned, we see this message increased at an early stage of neurodegeneration.
Okay, thanks, Osama. And maybe you can answer this one as well, Osama. They ask, have you looked at HD link in other neurodegenerative diseases?
Do I have to answer this question?
No, you can if you want, if you prefer
Yeah,
not.
no,
you can
we plan to do that. We plan to
Okay.
do that. Yes, we plan to do that. We think this is a very promising target, and Andrew sees that specifically in HD neurons in his model. But we haven't looked in other neurodegenerative diseases in the brains of people who died, say, from Alzheimer's disease or Parkinson's disease. We haven't systematically done this study, and I think it will be important to conduct.
Thanks.
I can only speculate based on what we have tried so far. There's a chance that HD link expression results from mutation within Huntington. It's testable idea. We can actually knock down Huntington and see whether this link RNA also goes down. So there's a chance that it's actually downstream of mutation within Huntington. And the reason why I'm saying that is because the first data set that we looked at after GSON identified HD link was late on Alzheimer's neurons, because we also have transcriptional data set for that. And in that context, we couldn't see it. So it could be something kind of at least partially specific for HD or mutation within Huntington.
Interesting. Here's a somewhat similar question. They ask if you have looked for HD link in peripheral HD tissue or if it's restricted to the brain.
So if we look at fibroblast, we may be able to detect some by qPCR and whatnot, but the expression is actually drastically increased when we turn them into neurons. So there's some degree of kind of upregulation in terms of cell identity.
Okay, thanks. So Megan Donaldson says, congratulations, Andrew and Osama, on an informative and hopeful talk. Do you have thoughts about what exactly HD link is doing to drive neurodegeneration, or is that for your future study? Have non-coding RNAs been detected in other neurodegenerative diseases? And if so, is there anything we can learn from that?
Yes, GSON right now has enough results that demonstrate that the pathogenic process or contribution of HD link to pathogenic process involves lipid dysregulation. So somehow it controls the amount of lipid accumulation, also lipid droplets in reprogrammed neurons. And it has a role in terms of controlling the dynamics of lipid accumulation in cells. In terms of other link RNAs in different neurodegenerative disorders? I believe so. There has been a couple of examples of how link RNAs may be differentially affected in primary tauopathy. In Alzheimer's, we also see in reprogrammed neurons, we also see different, not H link, but different link transcripts. So I think there's a lot to learn, but in terms of HD link, I think we have pretty good understanding of what's going on, and that a lot has to do with the lipid regulation.
Interesting. Thanks, Andrew. And I don't know if you guys are ready to talk about this because you didn't share it, but this person is asking if there are other long non-coding RNAs that are upregulated or dysregulated in HD, and if you've searched for other long non-coding RNAs, and if so, have you thought about targeting multiple long non-coding RNAs together rather than only focusing on HD link?
Always a great question. So this link HD link, we named the HD link because there's a gene annotation for it because it has been detected before in at least in cancer context. But there's another what we think is link RNA. We call this HINT. So it's a HD neuronal transcript. And the reason why we call it HINT is because there's no gene annotation for this the HINT transcript. So it's the way Jisun identified this link was because we don't know what other non-annotated genomic regions may be pronounced. The expression may be pronounced at least at the transcript level. And then Jisun did like sort of this kind of blinded kind of transcript hunting to look for any transcripts that could be specific for Huntington. So she found a couple, but she found this link. I think it's the link, but the HD hint to be very specific for neurons. So also they have to be neurons to express this. She also knows that if she tries to knock it down, it also cells are happier. So I think there's multiple modalities where we actually start thinking about all these non-protein coding transcripts that we can actually try to identify and target them at the same time. Yeah, it's a great question.
That's really, and maybe I'll add this is my own question too. And people are doing this in cancer, right? They're targeting long non-coding RNAs for cancer treatments. Am I correct in that?
Yeah, so there are some examples of that, and a lot of that has to do with the p53 regulation, the chromatin regulation, because
Yeah.
RNAs can be expressed inside the nucleus, and what they tend to do is they sort of interact or they promote the protein-protein interaction between different chromatin modifiers. So it actually has a kind of bigger role in terms of expressing or controlling gene expression patterns.
Interesting. So here's a question for you, Osama. People are asking if you're able to use tissue samples from live HD patients, and if so, how can they contribute to this? Do you have an answer of where they might be able to contribute skin samples or?
So where can they contribute skin samples? I think maybe Andrew can field that question because as our brain bank uh, preserves mostly brain tissue, we also preserve other tissue samples. However, there are specific ways that the tissue samples must be preserved in order for them to be utilized in a way that Andrew can use them. So perhaps Andrew, you can comment about the ongoing efforts that you guys have to expand that collection.
Yes. So in terms of using fibroblast samples to generate neurons, and then one aspect of my lab is trying to understand what it means to have neurons at different ages. So it will be great if we can actually do this in isogenic background, meaning that using neurons, reprogrammed neurons coming from the same person, but within a span of, let's say, 20 years. So for healthy individual fibroblast samples, we do have that. So there are some where people have collected fibroblast samples at once, typically during middle age, and about 20 years later, then there's another collection. So we call those longitudinal samples from healthy individuals, but we can still study the aging process using this method because each reprogrammed population from fibroblast represents a snapshot of that age. And then if we compare these neurons, isogenic neurons at different ages, we can actually see differences. We don't have similar fibroblast samples from the individuals with the Huntington's disease because the samples are not available. But if we have an access to such samples where fibroblast samples can be derived at different stages of the disorder, that experimentally, at least for experimentalists, that'll be a great resource that we can actually go after why sort of the adult onset, why there's a certain onset of the disorder or neurodegeneration onset when individuals reach a certain age.
Do you know if there's somewhere where people can donate those biopsies, or is there a researcher that's taking them, or should they reach out to their neurologist?
Yeah, I've been every chance I get, I ask around whether it's possible at all to collect such longitudinally collected fibroblast samples. So far, I have not been successful.
Okay.
I think, Sarah, there must be a concerted effort to create a repository, and maybe that is something that HDF can consider. And of course, I will be happy to work with anyone who will expand tissue-based research of Huntington disease. So I think that would be a great resource to create.
I agree. Thanks, guys. I like this question, Andrew, because this gets at your direct conversions versus iPSC differentiations. And they ask, do you find HD-Link and iPSC-derived neurons that presumably have not brought many of its epigenetic modifications with it?
Oh, great question. So we haven't compared side by side coming from the same fibroblast original samples. One way is iPSC versus direct conversion. But what we do know is that in terms of the aging, kind of being younger for iPSC neurons, what we do know is that if we compare sort of pre-symptomatic younger stage HD-MSNs versus kind of later stage symptomatic HD-MSNs, we do see differences in the amount of HD-Link. We can do that experiment where we basically try to modify the chromatin state to be younger, and then basically compare the expression between iPSC differentiated neurons and also direct reprogrammed neurons and see the difference. What we do know is that if we use the same HD fibroblasts, turn them into neurons, and go through this iPSC route, the neurodegeneration phenotype appears to be more pronounced in the direct reprogrammed neurons. I think in terms of asking, whether this one actually plays a role in neurodegeneration, we kind of have to stick to the direct conversion process because in that context we can measure the neurodegeneration phenotype.
Interesting. So here's another one that kind of gets at HD-Link levels. They ask if you've correlated HD-Link levels with repeat length in cultured neurons.
Oh yeah, so far in terms of reading the repeat length, so far we've been doing it in a sort of amateur way. We basically extract DNA, collect a bunch of cells and do PCR. But I think this kind of trying to measure this in reprogrammed neurons, we need more kind of more thorough way of doing that. So we are actually collaborating with Miriam Hyman's lab at MIT, trying to look at individual cells at a single cell resolution and see what population of cells actually have this repeat expansion. So once we do that, then we can try to see the amount of link RNA that can be whether there's a correlation or is it something that just happens with the starting the CAG repeat expansion length. These samples all have the repeat lengths in the range of 40 to 50. So it will be a good reference for us to see what population actually has the expansion.
Thanks. So this person kind of asks about translation of this. So they ask, would you see an HD treatment being something that potentially reduces HD link?
Osama, do you
Yeah, I mean, that's the reason why we're doing this study is to find cures.
Mhm.
We want to give people with HD hope that we can tackle this disease. We'll try our best using different approaches. As everyone here on the call knows, there are clinical trials to reduce the levels of Huntington protein, the Huntington transcript with antisense oligos. Um, once the results of this study show a conclusive benefit, which it appears that reducing HD link from Dr. Yu's work makes neurons happy, then it's a natural progression of that finding to try and translate this into clinical grade, perhaps. Of course, we have to go through all the preclinical studies that are necessary, but there is hope that we can get this into a stage where it can be tested. I think for the HD community, the more targets we have, the better chances we have at treating this disease.
That's great. Yeah, and you had mentioned, Andrew, about converting this into ASOs to target HD link. So that makes a nice transition for what modalities that we already have in the clinic. So maybe this is a good one to end on, but Osama, could you talk briefly about the importance of brain donation?
Yes, I can talk. I can talk, but I don't know about the briefly part. So, I mean, brain donation is, first of all, an eternal gift. When someone donates, um, when the family makes the decision, or the person with a disease like Huntington disease carrying the mutation makes the decision to donate their brains or even skin samples or blood samples, these specimens um, are preserved for decades, and they empower uh, hundreds of studies. So they make all the work that we're doing and other groups are doing, including, for example, Andrew mentioned Miriam Heyman's lab. She works with patient samples from donors who had Huntington disease. So it makes the research that empowers the finding of druggable targets and treatments for HD possible. Without it, we can't treat the disease. So it's not only about treating the disease. There's also the importance of doing foundational basic science that allows us to understand how the brain works. We can understand how the brain works by looking at mouse brains, primate brains, but the ultimate ground truth, if you may, is the human brain. So for a neuropathologist, I'm kind of biased in my answer, but I think it's critical. It's critical, and at Columbia we have a very large brain repository. We have more than 140 brain donations from people who died with Huntington disease, and this tissue is precious, and a lot of people need to use it. A lot of scientists need to use it for research, and we need more. We definitely need more.
Thanks, Osama. And if that's something that you're interested in, oh, go ahead, Andrew, you have a point.
so the quick comment I also like to add is because for experimentalists, when we decide to focus on some aspect of mechanism, we really need to confirm that this is something that's happening in a brain environment. So the more confirmation that we can have from neuropathological assessment, um, like what Osama does, it actually carries a lot of weight because now when we confirm that, it gives us the sort of drive so that we can that sort of inspires us to look into the mechanism.
Yeah, I agree. And so if that's something that you're interested in doing, there's lots of resources. There's the Brain Donor Project. You can go to their website. There's resources through HDSA, or if you're in Canada, Huntington Society of Canada, or email me if you need help in your local area finding a way to get a brain donation, because having that, as both Andrew and Osama have said, it is so, so critical to advance research, and it is truly the ultimate gift. So I think that's a fantastic way to end. We're at the end of our time, and so I'm going to wrap it up there. If you had a question that we didn't get to, I will send those over to Andrew and Osama. So if you left your contact information, they'll have a way to get a hold of you. I want to end by again thanking our sponsors: Ionis Pharmaceutical, Cleveland Clinic, Lou Ruvo Center for Brain Health, Novartis, and Argenta Therapeutics, as well as everyone who donates to the HDF to support the research that we fund. And of course, all of you for tuning in. And last but certainly not least, our speakers, Andrew and Osama, thank you so much for taking the time to tell us about this really exciting work.
Thank you.
Thank you.
Our next webinar is on Tuesday, November 18th. We are going to feature the work of 3 young investigators studying Huntington's disease. These are the future leaders of HD research, and registration for that webinar is now open. So after this, you can head over to the HDF website and get it on your calendars. We are so proud to showcase the advancements in HD research through our Research Spotlight webinars. So if you find these sessions helpful and you would like to support this mission, please consider donating today. Thank you for being a part of this community, and I hope to see you next month. Thanks, guys.